feel like I've got like 50 marbles in my head. So, viruses. There's a couple kind of interesting features on viruses that we'll kind of address, but in theory, you've got some of the kind of basic patterns. They're pathogens that can transfer or kind of exist by transferring their DNA, their genetic information, and getting it replicated by somebody else. Okay? The entire particle, we've got the viron, and then we look at the subcategories within it. It's made up of your capsid, your nucleocapsid, membrane envelope, and protein spikes. So all of those are kind of structural features used by the virus to propagate itself. And so we've got a picture that makes it look a little bit nicer or more violent, depending on your opinion. We have a membrane because it needs to protect. Okay? It needs to protect its genetic information. We've got these spikes on the outside. Those spikes are what it's using to then grab a hold of other cells so that it can inject its genetic information. Okay? So we've got the membrane to just kind of keep it contained. You've got its genetic information stored on the inside. That can be DNA, it could also be RNA. Okay? Depending on the type of virus, you're going to get different aspects. And so its general function or life cycle is it runs around, finds another cell, a healthy cell that has the material equipment to go through and process genetic information, sticks to that cell, dumps its genetic information into that, hijacks the transcription translation process, and makes that cell replicate itself. Okay? So the virus requires other things. Why it evolved like that, that's an interesting question, who knows. Okay? But it's kind of our very basic aspect. We've got genetic information. It now wants that genetic information propagated somehow, but it doesn't have the building blocks necessary to exist on its own. So it has to co-opt other cells that have all of those parts and pieces to do that replication process. So the first thing that they have to do is find healthy cells. So that's where the spikes come into play. They're looking for things that have those kind of markers that would suggest that that cell can replicate the virus. So it's got a bunch of receptors on the outside of it, on that membrane coat, that it then binds to or recognizes healthy cells. Right? And it can then attach to those healthy cells. Well, now that it's attached, it has to get its genetic information into that cell so that that cell starts to do its bidding. Okay? That's where the little spikes come in. It effectively injects, you can almost look at it as a needle, puncturing through the membrane and then spitting its genetic code into the host cell. Okay? That in and of itself is a pretty cool feature, but then we have to move on to the next aspect. And so one of the things we haven't talked about yet is genetic information. Well, that genetic code is now floating around inside the host cell. Okay? But typically our genetic information isn't just free-floating inside a cell, it's located in a nucleus. So that genetic information from the virus now has to somehow move through the cell and make it back to the nucleus where all of the parts to do genetic replication are located. So how it transitions from the outside all the way through the inside is still an area of active investigation, but a lot of it is coming back to, well, there's genetic information there, and our cells want genetic information in the central nucleus. So there's already a process built in to be like, oh, whoops, we've got some stuff floating around that shouldn't be out there, let's bring it across. Okay? So within the genes of our retrovirus, we have to code for a bunch of information. So we've got all of the basic pieces that we would need, the envelope, the coat proteins, and all of that. But the other one that can be potentially fun, depending on our virus, is we need something known as reverse transcriptase. Okay? And again, because we haven't talked about genetic information yet, okay, when we're looking at the genetic code, there are different processes for building genetic information. So we've got protein synthesis, which we can look at, and protein synthesis has to come from some kind of basic starting point. Well, where do proteins come from? What builds proteins? Let's go from, what's that? An enzyme. What's the enzyme that's building it? It could be another protein. Okay, well, if you're going to say it's another protein, we got a problem, because then I'm going to ask, well, how did that protein get built? Okay, so what's our original source? We could go all the way back to DNA. Is that true? Is DNA what makes proteins? No, what happens? We go from DNA to RNA, and then from RNA to a protein. Okay, so we have to move through each of those steps. And evolutionarily, it makes sense to be kind of moving through those independent steps, because the DNA can then be uber protected. Okay? Because if DNA was the one that was directly making the things that are now active and functional as proteins, that means we're reading the DNA a lot. Well, the more times you listen to, probably works better with VHS. Okay? If you remember your VHS tapes, okay? The more times you watch that movie, what ends up happening to the quality of the movie? It degrades over time and starts burning through. Same thing effectively happens with other things, but usually due to other processes, like you're taking your CD out and you scratched it. Okay? The more time we process stuff, the more degradation we get. So by having the DNA kind of separated out, okay, as a pure storage device, converting into RNA, RNA can now be my interpretation, the one that's actually doing the processing. And if RNA gets screwed up, oh well, I can just make more, because I've got the hard copy backup in the DNA. So when we're looking at our genetic information, getting all the way out to our proteins that are functional, we have to go from DNA to RNA to proteins. What do we call DNA to RNA? Transcription. From RNA to proteins? Translation. So it's looking at how we transcribe the information and then how we actually translate it out into the real world. So when we think about normal cells, we move from DNA to RNA to proteins, and we don't do the reverse. And we don't want to do the reverse because proteins get screwed up. And if proteins get screwed up and we do the reverse, well, then we'd be reverse translating bad information into the RNA. Well, and if the RNA is now more active, we're getting bad information in the RNA. If we reverse that, we're getting even more bad information into our source code. Make sense? So we don't really like the reverse process. We want to keep it DNA, RNA, proteins. Okay? But that's a normal cell. Viruses don't have all of the pieces to process. They don't have the enzymes to go DNA and RNA and proteins. They don't have all those parts, okay, for whatever reason. So they need to tell somebody else to do that. Okay? But the standard cell doesn't want the reverse process. So the standard cell doesn't have the pieces to do the reverse process, which means what does the virus have to supply? Those pieces to do the reverse process. That's our reverse transcriptase. Okay? So some types of viruses have a coding for a reverse transcriptase. And so what will end up happening is that protein will code for the process of taking its viral RNA and turning it into viral DNA. Now that it's viral DNA, that needs to make it into the host cell. Okay? So we have an initial reverse process, RNA to DNA, but now we also have to go viral DNA to host DNA. Well, that's also not really provided. So we need another enzyme supplied by the virus to allow that to happen. Right? So we get two big things that come in. We get our reverse transcription. So we get the injection. We get the reverse transcriptase, meaning our RNA, our viral RNA gets turned into double-stranded DNA. Okay? But now the DNA must make it into the host cell. So we have to take the viral stuff and integrate it into the host DNA. So we had a reverse transcriptase because it was reverse transcripting, and since it's an enzyme, we call it ACE. Okay? To take the viral DNA and insert it into the host DNA, we need to integrate it. We need an enzyme to do that, so we call it integrase, super creative. Okay? Once it's now integrated into the host DNA, the next time the host DNA goes through to replicate DNA, because we have the backup, but we have to constantly remake that backup every so often. The instant it goes in to replicate or transcribe, say, some information, I need another protein. I'm going to go back to my DNA source code, and I'm going to say, it's supposed to be this section is where that information is, so I'm going to read that section, and I'm going to make my RNA, which can then make my proteins. And it just so happens that that section is now filled with the viral DNA. So now the cell is end up getting tricked into producing all the pieces necessary for the virus to now replicate. Right? And then our virus gets replicated out and can then exist and do all sorts of fun stuff. So when I went through and looked at this, the thing that I thought was interesting was this integrase. I mean, reverse transcriptase is kind of cool too, but the idea of taking a foreign piece of DNA and inserting that into a host cell and how that process is done, I think is pretty neat. Okay, so I went and looked at the integrase protein, looked it up in the protein data bank, and looked for protein structures. And so when we go through and look at it, we've got this kind of model of these alpha helices, and we got all this purple stuff. What is all that purple stuff? It's not DNA. It's actually a bit weird. Okay. What they're actually showing here is an overlay of multiple models, or multiple crystal structures. Okay. Why are our alpha helices nice and neat, and then all the purple stuff looks like this massive, nasty mess? The alpha helices are consistent, which means whenever I get a crystal structure, I will always have the alpha helix shape. Okay? Because that's a standard stable form. Why are we seeing this big, massive, nested purple lines? That section is very mobile, which means when we look at the crystal structure for it, we end up getting different crystal formations for that section. Okay? And so what they're showing is the overlay of all of those different things. That overlay is going to be a big mess because it moves a lot, which then means the crystal structure isn't quite as clean there, and we end up seeing kind of a mess. Okay, so that is a multiple image overlay. It's not saying that that's what our protein structure is. That's a multiple image overlay. The part that's interesting on this is that they noticed that we had these alpha helices stack out in several different viruses the same way. So they had a repeating motif. Well, a repeating motif then gets called our secondary structure. And so the secondary structure, instead of now having alpha helices and beta sheets, and then we had beta barrels and all that other stuff, they said, well, there's a new binding domain. We will call that a zinc finger. Okay? So why they call it a zinc finger? Well, in that binding domain, what we end up noticing is that zinc is part of the crystal structure. Okay? So we have a new secondary structure based off of that. Okay? What's happening with the zinc finger? Well, it's those alpha helices that then fit inside the grooves of DNA. So it's the zinc finger literally grabbing a hold of DNA because the integrase has to be able to hold two different sections of DNA. So we've got this finger that goes in, grabs a hold of the DNA. Now the DNA is held, now it has the potential to do everything else it needs to do, which is cut it open and insert the new stuff. Okay? So it's just kind of a neat feature within it. We've got some nice kind of protein structure showing how those zinc fingers sit into the grooves of DNA. Okay? We also have the catalytic core. Okay? So that was just, the zinc finger was just grabbing a hold. Okay? But then it does have to do some chemistry. Well, that's going to be the catalytic core. And in that process, they noticed that, again, they had some repeating patterns, and they refer to this as our SH3 motif. Why did they pick SH3? I don't know, but that's what they did. Okay? And the SH3 has a lot of these kind of beta sheets / barrels, and those, again, are coordinating to the DNA, and that's how it has to work so that it can start to cleave open those DNA strands. So just kind of neat. We got some extra secondary structure pieces coming out of it, which is kind of why I thought I would address it. The mechanism of action of our integrase, it depends on the type of integrases. So you have two main types, serine and tyrosine integrases. And so what is ultimately happening there is that it's either serine or tyrosine that's involved in the actual catalysis of the breaking of the structure. Right? The serine is going to recognize short nucleotide phosphate sections, and tyrosine recognizes a longer section or a longer genetic code, gene sequence. Okay? The serine doesn't require a bunch of host cofactors, the tyrosine does. Okay? And so all that's really happening is it goes in, it recognizes a particular attachment site, and it initiates the cleavage of the backbone of our DNA structure. Once the backbone is broken, we can then start to pull it apart and put things in. Okay? So it's a neat little enzyme. Thought that was fun. And there we go. That wasn't too bad. That was roughly 15 minutes. And we'd have the exam questions. And so during the exam questions, we could say, well, let's just kind of go through these. Integrase is used by viruses to is to insert viral DNA into host DNA. Would it insert host DNA into viral DNA? No, but it's at least a nice swap around of the letters and numbers that it could then be a potential confusing answer. Okay. Viral DNA or viral RNA to DNA. That's not integrase. What was that? Reverse transcriptase. Okay. And then I thought that was a funny one, right? Determine the area in viral DNA. Like integrate the area. Yeah. So your answers don't all have to be awesome, but ideally there's some, at least, difficult aspect to a certain part. Which of the following is not a step in retrovirus infection? So somebody other than Jen. What's the first thing that has to happen? I would argue it's actually a step before A. Our virus has to bind to the outside of our host cell. Once the virus is bound to the outside of the host cell, what does it have to do? It has to inject the viral RNA into the cell. Once the viral RNA is in the cell, now what does it need to do? It wants to make it to the nucleus, but what's in the nucleus? DNA, so it can't get there. What does it have to do first? Become DNA. So it needs to be viral DNA. So we're going to have to convert the viral RNA to viral DNA. Cool, now we have viral DNA. That doesn't do anything until it's been integrated into the host DNA. Okay? So if we go through with the steps that we've got, inserting viral RNA into the cell, that's our earliest step here. We then need to transcribe our viral RNA into viral DNA, which is fun. That one's not even on there. Right? We then need to get the viral DNA into the host DNA, which should arguably be step 3. Actually, that's kind of bad wording. Come on, pen. We need to integrate the viral DNA into the host DNA. So now the question becomes between B and C, what's going on? So transcribe is referring to what? Replicate, and so this is where we have to be careful, and this is why this makes more sense to have after we've talked about the basics of genetics. So whoever's doing the genetic dogma really has a good lead-in to start this off. Okay, we start with DNA, we go to RNA, we have proteins. DNA to DNA is DNA to DNA. DNA to DNA. It's replication. I'm making the exact same thing. That's a replication. Okay, so we can't say replicate DNA to RNA because that's a problem. We're changing the nature. It's not a replication. We're converting that code into something else. Okay? We're converting that code into RNA. Well, DNA, RNA, what is the difference? Single strand versus double strand. Okay, the D is deoxy. Okay, so DNA and RNA are, for all intents and purposes, the same thing, except you removed an oxygen. Okay? So those people working on the structure of DNA can help out and explain some of that when we get to it. Okay? So DNA and RNA are effectively the same thing. So are we translating? No, you're transcribing. Okay? We're converting that from one person's handwriting to another person's handwriting. It's the doctor telling the scribe, make this drug, or not make this drug, insert this drug into the patient. But then what does the scribe have to do? It has to go find somebody else to get the drug, put it in. Okay? That's our kind of last step, and that's going to be our translation. So our transcription is DNA, RNA. Our DNA is replication. If it's viral DNA, and host DNA, it's not replication, it's not transcription, it's integration. You're taking the viral DNA and inserting it in. That's an integration. Okay? So if we look at our words, this is really going back to what our transcription is referring to. Our transcribing is RNA to DNA. We don't transcribe DNA to DNA. So our answer is...
But do you transcribe viral RNA to, doesn't it go viral RNA to viral DNA, not host DNA?
Oh, no. Yeah, you're right. That should be viral up there. B and C are both wrong. That's awesome. So which answer do you want it to be? Let's fix the other one.
C.
You want it to be C?
Yeah.
Okay, so let's make B correct. How would we make B, change the answer B to make sure it is now a step of retrovirus infection?
Let's just say viral DNA.
Make this viral DNA.
And C's wrong because that should say integrate, not transcript.
And yeah, to make C correct, it would have to be integrate. Does that make sense? So that version is what will be the final version. Realize just because we're using the same questions doesn't mean the same order of the answers is going to show up. So you can't just memorize answer choices. Make sense? Okay. So if that question shows up, I will make sure to fix that. Otherwise, you can laugh at me.
So these are fair game for the final?
These are fair game for the final. Yep. 3, retroviruses store their genetic information in B in our RNA. Okay, that's the retro part. The retro is saying the information is stored in something other than DNA. Okay, because you can have a virus have that information stored in DNA. Retrovirus is stored in RNA. True, false. Viruses are always deadly. False. They don't necessarily have to do anything damaging. The following are all secondary or tertiary protein structures, except for. They're all valid. All of those are secondary/tertiary protein structures.
And these questions, are you going to post them?
Yep,
Okay.
they're officially already posted, even with that nice little typo in too. What I will try to do is copy and paste everybody's exam questions out into a single document and then post that. But you have to give me your questions in a reasonable format. So if it takes me forever to format it, then I'm not giving you a copy of everybody.
Is this
You
okay,
have to
or
deal
do
with
you
it.
want
This is fine. This one's a little bit messy because of how I got those questions on there, but I could get it out. A Word document is easier. Make sense? For those who's keeping track, since I did record this, that was 25 minutes. I would be yelling at you to stop 10 minutes ago. Okay?
25 minutes
with all the questions in, yeah. And I think we started recording a little bit early, but yeah, makes sense? Okay. Lipids. Yay, lipids. Biological membranes and lipids. So, I don't know why I didn't think of this. Like, close. So it's kind of what's going on with our lipids. So big words that come out of this. Amphipathic. Amphipathic is referring to the ability to interact both with water and with hydrophobic species. So it's a hydrophilic and hydrophobic thing. Okay, which means we'd have to go all the way back to intermolecular forces. And because our instructor for the class did this awesome little video or thing where they showed pictures and colors and intermolecular forces, I was like, that's a good thing to show, to get like brownie points or something. Okay. So kind of the point behind lipids, or this section of lipids, is to get both at the structure of what are lipids and what they're useful for. Okay? And so when we talked about membranes earlier on, we also had this fun little picture showing bilayer sheets and liposomes and micelles, and we said that those were built off of lipids. Okay? And we said they had the hydrophilic tail and hydrophilic head and the hydrophobic tail. And that's how we generated all of our structures. So what are they? Okay. So it's really just a class of biochemical molecules that have this amphiphilic characteristics. And so it's really going back to solubility. So we're looking at any kind of molecule that has this solubility characteristic, where we have a hydrophobic part and a hydrophilic part. Okay. And there's lots of them. Right? We have two main classes. We have what are known as open chain, because it's just a carbon chain. Right? It's just a long structure with just a whole bunch of carbons, and at one end we have the classic example, our carboxylate. Okay? Which I don't have the classic example there, but it is on the next few slides. Bunch of carbons, and then O minus. Okay? Also known as soap. Okay? Soap is an open chain lipid. So we have that polar head group and the nonpolar tail. So we've got all of these different kinds of lipids. So fatty acids, and I don't even want to list them all off, but there's a bunch of them. Okay? We then also have cyclic versions. Okay? A lot of the hormones that we work with tend to be lipids. Okay? Some of the more important things, cholesterol is a lipid, and we've got a nice structure there. Notice cholesterol has a whole bunch of carbons and hydrogens, and if we look through that whole structure, we've got a bunch of those. And that would suggest it's very, very hydrophobic. Okay? And I always thought this was kind of a weird one to call a lipid, because I'd say all of that, it says it's hydrophobic. Where's the hydrophilic part? The one alcohol is enough to make this considered a lipid. Okay? We have a small part of that structure that is now hydrophilic, and we have a massive part of that that is hydrophobic. Okay? Because of that, it's then classified into our lipid section, and because it's now cyclic, we have a bunch of rings in it as opposed to the straight chain, we call it a cyclic lipid. Might have heard testosterone, kind of common progesterone. So common hormones, and if we compare those hormone structures, they're all derivatives of cholesterol. Okay. They do still have some of that amphipathic material. It's not quite as neat, because if we go through and take a look, for instance, at testosterone, we still have tons of carbons and hydrogens through the middle, but we have kind of a polar at one end, and we have a polar at the other end. So it has a little bit less of that amphipathic nature, because each end is polar. Okay. But again, because it has some origins back to our lipids or cholesterol, they get grouped in. There it goes. So fatty acids are the most common one. So we've got a bunch of fatty acid structures drawn over here. Notice the variation between these is double bonds. So we've got some and chain length, number of carbons in it. So when we're looking at our fatty acids, we're typically looking at the number of carbons associated with it, and it usually falls in this 12 to 20 range. Okay? Much less than that, it doesn't get used as a fatty acid as much. It's in kind of its processing stage, getting moved into biosynthesis. The next part is then referencing some bit of nomenclature. You'll sometimes see reference to, say, like a 12,2 fatty acid. Well, what the heck does a 12,2? Well, the 12, the first number is the number of carbons in my chain. The 2 is the number of pi. The pi being the pi bonds, the double bonds within it. Okay? So a 12,2 is now a 12-chain fatty acid with 2 pi bonds. If I want to add even more information to that, I can then specify where that pi bond is located using the symbol delta followed by the number as a superscript. Okay? Why did they pick that? I don't know. Because they're weird. So delta 5 or delta 5, 7, I now have a pi bond at positions 5 and 7. Pi bonds, particularly pi bonds in a long chain, are also going to have the added nomenclature bonus, if you will, of being cis and trans. Okay. Will the cis and trans have an effect on the structure? Yeah, it has a massive effect on the structure. So when we're trying to name them, we'll almost always reference them as cis and trans. Right? If you think back to just kind of like health crazes, say, oh, trans fatty acids, those are bad for you. Okay? Why would trans fatty acids be bad for you and cis be good for you? Okay, well, some of that is coming down to kind of the origins of where these fatty acids come from. Okay? Fatty acids are in both animals and plants. Okay? Everybody needs membranes, so we have them. Okay? In animals, we tend to call them fats, and in plants, we tend to call them oils. Why would we do that? When you think fat, what do you think of? Solids. And when we think oil, we think liquids, which I think is kind of a fascinating concept, because if I looked at plants and animals, okay, I would say animals move and plants don't. So what would I expect between fats and oils? I would expect plants to be fats because they don't move, and yet they're actually oils. Right? It seems kind of like an odd concept on what's going on there, but it has to do with what other things animals contribute into their membranes to change some of that kind of mobility. Okay? The next part comes into our saturated versus unsaturated. Saturated means no double bonds. Unsaturated means we have double bonds. Okay? Well, how would that tie back to our plants versus animals? Okay? Well, our fats, are they saturated or unsaturated? Yeah, animals are typically saturated fats. Okay? And plants are unsaturated. Okay? How is that going to change our phases of solids and liquids? Well, if I take a look at, say, a saturated fat, the middle one, okay, and I compare that to an unsaturated, double bond up there, how is that going to change how those molecules can stack? The unsaturated one, because of all those kinks, now can't stack as nice. Well, if it can't stack as nice, the intermolecular forces between those are now weaker. With it being a weaker intermolecular force, it requires less energy to cause the phase transition, which is why our unsaturated fats tend to be more oily, or liquids. Okay? And our saturated ones tend to be fats, okay, because they can stack neatly. So one of the things that we could go through and do is we then reference this idea of trans fatty acids. Well, trans fatty acids, if we think of trans, when do we see the trans fatty acids pop up? Anybody know phases on those? Solid, liquid, gas? Yeah, they tend to be solids. Okay, why would the trans fatty acids tend to be solids? They're what? they're still unsaturated because they have a double bond. Okay, so let's take again a look at that middle one, and let's make that a trans fatty acid. Add a double bond there. That one would be trans, right? Did that change the structure? No, it's that nice, still flat, straight chain. That can now interact really, really strongly, and I'd get more of a solid phase. So if we look at our plants that are oils, one of the things that we kind of went through and did is, oh, well, butter, animal fat, oh, that's bad, that's dangerous, we don't want that. Oh, but I like that it's spreadable. I can cut butter and I can spread that on something. Okay? And then having all those double bonds, oils and plant fats, oh, those are healthy ones. So I want those, but I want to be able to spread them. Okay? Well, I can't spread them if they're the regular oils with all those cis double bonds. So what do? I make them into trans double bonds. Well, now it's healthy because they still have double bonds. It's not the double bonds that make it healthy. It's that it's not solid. Okay? It has to do with how those things end up stacking and interacting. Okay? So when we went through this process of, oh, I'm going to make now healthy fats by hydrogenating plant oils. This was the whole fad with margarine. We said, oh, plant oils are better. Well, we just added a bunch of hydrogen to our plant oils to then make them saturated fats. But because they came from a plant, they're now healthy, even though structurally they're identical to the animal fat. Okay? Because we limited some of that hydrogenation, that's where you get trans fatty acids coming in. It's because you've hydrogenated. You added hydrogen to remove the double bond. That usually requires heat. Heat gives enough thermal energy to our fatty acid that eventually the cis double bond can rotate to the lower energy trans double bond. Okay? And that's why when we heat plant oils, we tend to start losing the cis fatty acids and we start getting the trans fatty acids out of it because the trans form is lower energy. Steric hindrance and all that fun stuff. Okay. And those of you counting, I think that was 15 minutes. Just so you know. So, storing fatty acids, plants tend to store them as waxes, okay, or really they don't really store them as waxes. They use the waxes as a coating to help protect because then that repels water. Okay? It repels water because when we look at it, we've got our fatty acid at kind of to the left on both of these. There's our fatty acid part. Okay? Well, we've got the polar head group, and then we have the non-polar fatty part over there. What did we do to make it a wax? We added that section, which was a bunch of carbon hydrogens. That's all non-polar. So what happened to the polar part? It's now trapped in the middle. We don't have a polar part anymore, which means if I now put this as a coating on a surface, there's no interaction with water, and it'll actually repel water away. So we can use that as a protective layer to repel things off of it. Okay? The most common place to store our fatty acids in something known as triacylglycerols, or more commonly, triacylglycerides, okay, or triglycerides. Okay? What those are referencing is we take a fatty acid and we take a glycerol. Glycerol has 3 reactive alcohol points. Those alcohols can act as nucleophiles, attack our carbonyl, kick out water as a leaving group through a sequence of organic chemistry steps, which we don't need to stress about, and we now have that fatty acid connected to the glycerol. Okay? If I do that a whole bunch of times, I then have a triacylglyceride. Okay? And that I can now use as a more complex lipid. And that gives me energy storage because now I've got a whole bunch of those carbons there. I could start to eat some of those carbons off to release some of that energy. Okay? So that's our base structure. Well, then we end up with phosphoacylglycerols. Well, didn't we have acylglycerol before? Yeah. So now it's just phosphoacylglycerol, meaning It's the exact same thing except I put a phosphate on it. Right? Is that going to change the intermolecular forces? Well, yeah. Now the phosphate group is ionic. I'm going to have a very polar head group because of that phosphate. I'll still have my nonpolar tail, but I now have a polar head group. Cool. Okay? So that can change some of my intermolecular forces with this. Okay? Just because fatty acids are irritating, this one's known as a phosphatic acid because we have just the phosphate attached. Okay? And it's the acid because we have that H. What happens if that H is a carbon? Well, I can't call it an acid anymore. Okay, what kind of bond would I have?
Ester.
Yeah, it's a phosphoester because it's phosphorus being involved in it, and it ends up becoming phosphatidyl ester. Okay? Because it's now an ester, we've changed the structure. Again, that means we have a new fatty acid, and we have to come up with a new name. Along those lines, I've lost the connection. We end up with things called sphingolipids. Well, what the heck's a sphingolipid? We're taking our glycerol molecule and we modify the glycerol molecule. Okay? We modify it so we have this alkene at the top end of it, a large nonpolar part. Okay? We still have the connections for fatty acids. Okay? Because it's still a fatty acid store thing, we now have to come up with a new name because it's now not officially a triacylglyceride. It's got a different piece to it. Okay? The piece that we added was what's known as sphingosine. That's replacing our glycerol. Okay? So we have a sphingolipid because instead of glycerol, it's sphingosine. What if we switch that up and we used a ceramide? Well, that's our triacylglycerols. We end up with a whole myriad of different names coming from our lipids because we're changing the base piece that holds on to the fatty acids. You change that base piece, you get an entirely different, not entirely different, but different structure, which then means different functions, different names associated with it. Okay? Again, what happens if we went through and added a carbohydrate? Well, now I can't call it a sphingosine, I can't call it a triacylglyceride, I'm going to have to call it a glycolipid because I have the lipid part that was the fatty acid, but now I've also added a carbohydrate, glucose, so now I'm going to call it a glucolipid. Okay? So it's turned into kind of like a Frankenstein of nomenclature. Okay? Oh, I had this new piece and this piece, so I'm just going to call it a new thing that's bits of each of those pieces. Okay? And yeah, that can kind of continue on almost indefinitely. Okay? What happens is we then can insert these into membranes, and that's how we get our overall structures. Plants use unsaturated fatty acids, which means they're a little bit more fluid, okay? Which is an interesting feature. Animals are less fluid because they use saturated fats. We still need it to move, so we have to insert things that disrupt that strong interaction. So we insert things like cholesterol, which is that big ring structure which now disrupts some of those intermolecular forces and allows a little bit more mobility behind it. Okay? But I thought plants were rigid. How are plants rigid? They have another membrane. They have cell walls. Okay? The biggest thing I really want to get at here is our membrane composition. So if we go through and take a look at a membrane, we saw like a really easy versions like this, okay? Where we got those nice little tails, little circle thing, and that's kind of the end of it. Okay? Really what we're doing is taking all of our different lipids, and those are now getting integrated into our membranes. And we have varying amounts of each. Okay? Why do we have varying amounts? Well, that changes the intermolecular forces at the surface. Do I want different forces on the surface of a membrane? Well, if part of that cell is aimed one direction and say it needs to release a bunch of ions so that the next cell knows that ions were released so that we can then propagate a signal from where you put your hand on the burner to get to your brain to say, hey, you should stop doing that. Okay? There's a directionality associated with that. That directionality can come from what we've put in the membranes. Okay? other parts within this. If we look at the inside, notice the inner ones are almost all phosphoacylglycerols, and the outer ones, we have a whole bunch of variation. Why would we have more variation on the outer ones? Those outer ones, sphingomyelin, cerebroside, our gangliosides, what is special about those in comparison to the phosphoacylglycerol? Big. Okay. Sphingomyelin, the cerebroside, the not phosphoacylglycerols are bigger. Okay. Why would that matter? They can interact on the outside of the cell wall. You're looking at the curvature of the cell. Okay. Being on the outside of the cell, we need to fill out a larger surface area. We're going to have larger pieces. The inside, if we put in one of those larger pieces, what happens? We're going to increase the spacing between those, and we change the porosity of our cell membrane. Okay? So the inside tends to be smaller and more compact to kind of keep things trapped on the inside. The outside will be a little bit larger so that different things can interact and potentially move through it. Okay? Membrane fluidity, this goes back to how we stack things. We kind of already talked about that. Really what I want to look at is our model of our membranes. We have transport across the membrane. So we'll have proteins bound in it. Those proteins facilitate whatever they were designed to do. Almost all the citric acid cycle is happening within the membrane. Okay? Electron transport, all of those are membrane-bound proteins. Okay? And they're facilitating transfer across or otherwise. Okay? So we have integral proteins because they're integrated into the membrane, and we have peripheral proteins because they're on the surface of the membrane, not embedded in it. Okay. How do we get almost there? Fluid mosaic, this I thought was just kind of neat. Okay. What someone went through and did is they took cells, they froze them real quickly, did a flash freeze, and they could crack them open. And in the process of cracking them open, they could crack them along the cell membrane, meaning we could then see the inside of our cell membrane. So parts of it, we'd see these little holes, those holes because, well, protein was sitting there. On the other side, we see all these little pock marks where there were the proteins themselves. And sure enough, that ends up being what we see. We don't see this big smooth surface. Look at that. I mean, that's nasty. All those little, like zit things popping out of our membrane. Okay? There's a lot of stuff in the membrane. Okay? Lots of proteins are there facilitating transfer across. Okay? So it's not just this smooth, oh, it's just a membrane. There's a crap ton of stuff within that facilitating transport across and different reactions. Which gets us kind of to our last part, passive versus active transport. Passive just means it moves across on its own. Active means we have to force it across. How do we force something across? Energy. That's where we bring in our ATP. Okay? So our passive comes back to concentration differences, like in ATP synthase. We have a passive transport of hydrogen across the membrane to generate ATP. How did we get the hydrogen across the membrane to begin with? Well, that was an active transport through our electron transport chain. Okay? So the cell has worked out in different situations to run different things. Okay? Simple diffusion is our passive one, just kind of freely moves through. Facilitated diffusion, things can get carried across with something else. Okay? So you've got a chaperone that carries with it, but it's not requiring energy to move through. Our active transport drives against gradients. This happens a lot within muscles. We're getting ions moving back and forth across membranes to cause contraction or not. Okay? You're usually using ATP, and we can do secondary kind of transport using proton gradients. Vitamins. Vitamins are lipid soluble as well. Ta-da, vitamins. Exam questions. So yeah, that one was, geez, like 25 minutes, 30 minutes. Yeah. Not counting the questions. Yeah. So, just to give you an idea, that was 15 slides. Okay. So, exam questions. Which of the following is not an example of a lipid? D, amino acids are not lipids. Okay. Why is margarine more liquid than butter? Okay. What is margarine? That can be helpful to know. That's hydrogenated. Hydrogenated what? Unsaturated fat, and that's where we run into an issue. Fat is typically associated with animals. Margarine is typically not animal, it's plant. So we're looking at hydrogenated oils officially. Okay, so we're adding hydrogen to our oil. So why is hydrogenated oil still more liquid than butter? It is partially with plants. It has a larger percentage of unsaturated fat. We have the double bonds in there. Okay? Just because it comes from plants, and even that is questionable. Hydrogenated oil does not come from plants. Hydrogenated oil comes from humans that took plant oil and did stuff to it. So it's not coming from plants. Which of the following is not a characteristic of passive membrane transport? Man, I'm a big fan of the knots this time, aren't I? So not. With the concentration gradient, well, that is passive by definition. ATP from ATP synthase. Man, that's kind of nasty. Oh yeah, that's fun. Let's call that D. Typos all over the place. Simple diffusion is passive. Okay? And we've got kind of the example that we already talked about. ATP from ATP synthase. How did that form? That was transport of hydrogen across the membrane. What does ETC do? ETC is our electron transport chain. That's also the transport of hydrogen across a membrane. ETC is forcing hydrogen across. It's establishing the gradient. Once the gradient is established, then it will slowly work its way back through a passive transport mechanism in the formation of ATP. Okay? So ATP is still a passive transport. ETC is the active one. So ETC is not a characteristic of passive. It's active. Oh, I didn't number these. That was nice. Top one. Cholesterol is not found in which of the following? They do have good fats, but not cholesterol. I think the only reason I asked that question is because I had a high school teacher that was like his favorite story. He kept playing that whenever he was in college or something like that, some teacher said that avocados had cholesterol in them, and he was like, "No, they don't." And he called the teacher out, and then the teacher then asked the class, "Everybody's already heard this before, right?" And he was like, "Well, yeah, you just told them, dumbass." So yeah, he got kicked out of that class. Sphingomyelin has a relatively larger polar head than phosphoacyl or phosphoglycerol. Where would sphingomyelin be found in a small cell? Okay. So we're definitely referencing our membrane. Where on the membrane? So inside the membrane, that doesn't really make sense. That's just saying inside the cell. That's the cytosol. So B and D are the same thing. Looking at the face of our membrane. On the inside of the membrane, because it's a small cell, I need tiny head groups. The larger those head groups, I wouldn't be able to shrink down the size of the cell. Okay, which means if sphingomyelin is big, it better be on the outside. So we're looking at the exterior face of the membrane. Make sense? Questions about all that? And look at that. A whole 4 minutes early.